Let's talk about nutrient digestion and metabolism. In this talk, we're going to cover how different nutrients are enzymatically digested. We'll cover the process of absorption. We'll cover the basic concepts about nutrient digestibility. And we'll talk about why we use bioavailability for trace minerals as well as vitamins. And we'll briefly touch on nutrient metabolism. First, let's look at enzymatic digestion. An enzyme is a protein that specifically catalyzes a biochemical reaction. An enzyme is a protein. It could be other molecules, but generally speaking, it's a protein. And its nature is basically to lower the activation activity of a specific biochemical reaction, therefore making the reaction easier to occur. Enzymes are highly selective of their substrates. For example, we're looking at a phytase enzyme, and its substrate is the molecule of phytate. This phytase enzyme is not going to act most likely on any other substrate other than a phytate. The activity of enzymes is highly regulated by the environmental conditions, and that also includes the substrate concentration as well as the concentration of the products that will result from this reaction, and cofactors, coenzymes, inhibitors, pH, and temperature. Like we were looking in the example of this phytase reaction, we have phytate in water, and the product of this reaction is phosphate plus the myo-inositol molecule minus a phosphate. But notice here that in principle there is no other chemical compound produced other than phosphate in this particular example. The general mode of action of all the enzymes is that they will act upon a substrate to produce a product or products. The enzyme is neither part of the substrate nor the products. It simply will catalyze this chemical reaction. More specifically, for example, an amylase enzyme can work on starch to produce glucose monomers. In the case of a protease will act upon a protein or proteins, and the resulting product or products will be peptides and amino acids, depending on the type of protease and the type of protein that is acting. There are specialized enzymes for each type of substrate. If we look at the feed, we can divide the feed into dry matter and water. Water is a nutrient that does not need to be digested, and it's simply going to be absorbed. The dry matter, we can divide it into an organic and an inorganic fraction, and we can further start to break down these into smaller and smaller compounds. We can talk about nitrogenous compounds, carbohydrates, phenolic compounds, lipids, vitamins, and other minerals. And furthermore, we can break it down even more into protein and non-protein molecules, starch and structural carbohydrates or fiber, lignin, tannins. The lipids could be simple, could be complex. Vitamins could be water-soluble, lipid-soluble, microminerals, etc. The important portion here is that for each one of these great categories that we have here, we do have a specialized type of enzymes or enzyme family. For example, proteases will act on protein, amylases on starch, lipases on lipids, and these are some of the main enzymes that are produced endogenously in the pig. There is a group of enzymes exogenous, which means do not belong to the pig, but that we can add to the feed to improve the digestive process. That includes several fiber digesting enzymes like beta-mannanases, xylanases, glucanases, and more, as well as phytase, which we briefly touched before. And these are enzymes normally of microbial origin that are added to the diets of pigs in order to help them digest some of these compounds or to reduce the anti-nutritional effect of some of these compounds. But the bottom line is that for every type of substrate, there is a specific type of enzyme. If we look at the digestion and absorption of lipids in a little bit more detail, we'll find out that these lipids are really come in the diet in the form of triglycerides. The lipase enzyme, what it does is free the fatty acid in position 1 and 3, then absorption takes place, and then that triglyceride is reassembled inside the enterocyte. From there, it can go into the portal vein, into chylomicrons, and other type of transport vessels. From there to the liver, and from the liver to the rest of the body. There's a lot of different steps in the metabolism of fatty acids and the transport of fatty acids, which we're not going to go into detail in this talk. In the previous slide, we talked about digestion of triglycerides. And yes, there's many different types of fatty acids, but the molecule that gets digested is a triglyceride. When we get to carbohydrates, we see that there is a little bit more diversity in terms of substrates, because we can have polysaccharides, dextrins, sucrose, maltose, lactose, and other carbohydrates. Each one of these type of substrates requires specialized enzymes like amylases, maltases, or lactases. The end result of most of these carbohydrate digestion is the result of monosaccharides like glucose, galactose, and fructose. We know that there is different and very complex mechanisms, which we're not going to go a lot into detail into this talk, but mainly to indicate that glucose is absorbed mainly on a sodium-dependent manner, whereas fructose is not absorbed in that way, and it does not require the co-transportation of sodium. Once carbohydrates are absorbed, then there is a lot of complexity as to how that primarily, let's say, glucose appears in blood, goes to the liver. The liver takes a portion of it to synthesize glycogen that will be used later on to regulate blood glucose levels. How that glucose increase in blood produces an increase in insulin, and how that insulin stimulates tissues to uptake that glucose from blood. There's a lot of metabolic processes that take place, and then I just want to illustrate a few points in here, but we're not going to go in this talk into full detail in terms of carbohydrate metabolism. When we get to the digestion of protein, it's where the complexity of digestive process really kicks into high gear. We know the digestion of proteins will start in the stomach by the secretion of hydrochloric acid to lower the pH. The reason why the pH is lower, yes, it has a component to try to deter the entrance of pathogens, but the main purpose is to provide a low enough pH for the pepsin to be active. And what these pepsins are going to do is they're going to start acting on the different dietary proteins, and they're going to start breaking down into smaller and smaller peptides, and hence the beginning of the protein digestion will take place, which is the one that is probably extended the most and the one that is most compartmentalized along the gastrointestinal tract. From the stomach, we move to the small intestine, starting at the duodenum. And once the acidified digester from the stomach is received, then it's going to be neutralized by secretions of bicarbonate from the duodenum as well as the pancreas. And then again, the reason for this change in pH is to provide an environment where the enzymes, mainly from the exocrine pancreas, can work. These enzymes are secreted, as we mentioned previously in a talk, as zymogens or inactive proteins. And these inactive proteins are going to be activated once they reach the lumen of the intestine. This process is first carried out by an enzyme that is secreted by the duodenal mucosal cells, and this enzyme is called enterokinase. And this enterokinase will convert inactive trypsin, which is called trypsinogen, into active trypsin enzyme. Once the trypsin is active, it can autocatalyze itself, and trypsin is able to act on trypsinogen in order to produce more trypsin. Now, this trypsin enzyme also is going to act on all the other zymogens, and then it's going to cleave all these zymogens or proenzymes and literally are going to transform them from inactive molecules to active proteases. And this is one of the main reasons why we're so concerned with trypsin inhibitors. It's not that trypsin is such crucial in terms of actually all the work that it does by itself in terms of protein digestion, but it's because of its highly critical role in activating all the proteases that come from the exocrine pancreas. Therefore, if we have the presence of trypsin inhibitors, and if we reduce the activity of the trypsin enzyme, by default, we're reducing the activity of all the proteases that are secreted by the exocrine pancreas. These proteases, like the elastase and the carboxypeptidase and the chymotrypsins and the trypsins, they will have the function of taking the peptides and the proteins that have been broken down in the stomach and continue to break them down into smaller and smaller peptides. The third big step in protein digestion, it's the brush border. And here we have, again, more proteases are secreted. And the main objective here is the same as in the stomach and with the proteases from the exocrine pancreas. It's to simply reduce the molecular size of the proteins until we get them into the three main forms that are absorbable. Those are free amino acids, dipeptides, and tripeptides. And then there are different absorption mechanisms for both free amino acids and small peptides. In the case of free amino acid transporters, we have two great families. One is a sodium-dependent family, and the other one is a sodium-independent family. You have all the different systems, whether they're sodium-dependent or sodium-independent. And all I want you to take from here is the appreciation that on this slide we have 17 different amino acid transporters. So at least 17 different types of mechanisms to absorb different types of amino acids. So you can see the complexity is pretty great, and that's why protein digestion and absorption, we spend a little bit more time compared to carbohydrates and lipids. We mentioned that the digestive tract is able to absorb not only free amino acids, but also peptides, mainly in the form of di and tripeptides, where these are molecules of 2 and 3 amino acids. And again, there's different mechanisms. Some of them are sodium dependent, and then it requires energy as well. The other thing that I want you to know from these graphs is that the absorption of amino acids, it appears to be more efficient when these amino acids are present in a small peptide form compared to when they're present in the free form. And in this example here, we can see how the absorption of glycine, when it's present in the dipeptide glycyl proline, it's much higher than the absorption of glycine by itself. Once the amino acids are absorbed, they literally go into the enterocyte. In the enterocyte, we're going to have metabolic processes going on. And this is a very interesting portable blood appearance balance. And these numbers are expressed relative to the intake of each one of these amino acids. As we can see, most of these amino acids are not appearing in a portal blood at a rate of, let's say, the expected 100%, which will be ideal. Most of them are below, which will indicate that the digestion and digestibility is not 100%, which is actually biological, very accurate. 2 points here worth noticing. One is glutamine, and glutamine has a net portal appearance rate that is negative. How is this possible? This is possible because the enterocyte, basically, it's using all the glutamine that is coming from the lumen of the intestine from the digestion process, as well as all the glutamine that is coming via the mesenteric artery for its own metabolic purposes. And that's why we have a net appearance that is negative. Now we look at the other extreme, and we look here how alanine has a net portal appearance of about 250%. the intake of alanine. So what is going on here? What's going on here is within the enterocyte, there is a lot of transamination process and deamination and transamination process. This basically means there are a lot of those amino acids that are broken down like glutamine, and then we have that excess nitrogen that need to be placed on something, and that something, it's going to be very likely a pyruvate. Pyruvate plus an ammonia group will give us alanine, and that's why we see alanine being secreted into the portal blood at a much higher rate. And this is basically pyruvate and alanine are functioning here as a shuttling system from nitrogen from the enterocyte into the blood to the rest of the body. We have seen this slide before, and we're going to see this slide many times over these talks. And again, what I want to emphasize here is that those proteins that are not digested and those amino acids that are not absorbed, eventually they're going to encounter a high population of microbiota, and this microbiota is going to carry on the process of fermentation. Fermentation of proteins in general terms is something not good for the animal. Why? One of the main reasons, like we talk about, is because it produces compounds that are not helpful to the animal. They could actually be negative for growth, and they could actually alter the microbiota. We've also seen this slide before, and we're going to see it several times more. Again, in this study published more than 40 years ago, what the researchers are trying to do is are trying to link and establish the relationship that when protein fermentation is high, growth is low. And when protein fermentation is low, then growth performance is high. We can debate here whether that growth performance is high or low because the pig is able to digest more or less protein, and because it's able to digest more or less protein, or because it's able to ferment more or less protein. The reality probably is that both processes are linked. If the pig digests and absorbs more protein, then we're going to have less of a nutrient flow. We're going to have a lower concentration of substrate protein in the cecum, in the column for fermentation. On the other hand, if the pig is digesting less protein, then the amount of substrate protein that is going to flow into the cecum is going to be greater. And like we saw, one of the factors that affect the mode of action of enzymes is substrate. Protein, microbial protein fermentation, it's run as well using enzymes, and those enzymes are also substrate dependent. Like we mentioned before, the solution is not necessarily to reduce protein levels, because animals do require amino acids. And then, although the animals does not require protein, it requires amino acids, that protein is the source of amino acids. So we can see that as we increase the amount of protein in the diet, we get a better growth response of animals. So here, the question is, how do we strike a balance between providing enough protein and amino acids to the pig without providing excess that could be fermented and be deleterious for the intestinal health and the growth performance of pigs? I will illustrate again the relationship between digestion, absorption, and fermentation. So the feed that the pig is consuming will come in contact with enzymes that we're here in this talk in a little bit more details. And those enzymes will break the feed into smaller and smaller compounds until some of those compounds eventually are small enough that can be digested and absorbed. Some of those cannot be digested any further for a variety of different reasons, and those undigested portions of the feed are going to encounter the microbiota, which are going to carry along the microbial fermentation process, which also involves a variety of different enzymes, and is going to give us a variety of different products, whether we're talking about fermentation of carbohydrates or whether we're talking about fermentation of proteins. And those are the two primary substrate sources that are going to be fermented in the hindgut of the pig. And like we mentioned before, the fermentation process can be very useful to the pig, like volatile fatty acids or short-chain fatty acids, but others, like we saw in the previous slide, like paracrisol, could be quite harmful. Let's review what we have talked about and discussed thus far. What is the main source of trypsin inhibitors in the diet of pigs? It's soybean meal, or in general, soybean products. And that's why we have to put attention to the quality of those products, and we'll discuss that more in detail in other talks. We can say that amino acids are mainly absorbed as what? Amino acids are mainly absorbed as peptides of 2 or 3 units, or as we call dipeptides and tripeptides, although there's also a variety and a good amount and an important amount of amino acids that are absorbed as free amino acids. And then the last thing is to say, remember, in the digestive tract, everything that is not digested and is not absorbed is subjective to fermentation.